Now that we've had an introduction to the endocrine system, an important control system of the body, let's take a further look at this important system. We said in the first section that the degree to which target cells respond or change what they're doing in response to a hormone depends on the number of activated or hormone-bound receptors per cell. Now there's three things that are going to impact that value, that number of bound receptors. The affinity, the tightness of binding of the hormone and the receptor. The relative number of receptors on a given target cell. There's a lot more receptors, there's potential for a lot more bound receptors, activated receptors. And finally, the blood levels of the hormone. What's the concentration of hormone coming to those receptors? So the first of those three, affinity, means grip or tightness of binding. Here's something interesting about hormones and their receptors. Like any chemical signal, they bind to their receptor, but it's not a permanent binding. There's a binding and then dissociation. Binding and dissociation constantly happening. And so some of the receptors are always bound by hormone if it's present, some of them are not. And the question is how many. That determines the activation of the cells. The higher the affinity, the more time the hormone will spend bound to the receptor, and therefore the more activity will be produced. The number of receptors per target cell. Well, with respect to that, there's some things that happen. There's some dynamic things that happen. A given population of target cells will increase the number of receptors on their surface when hormone levels go down. They kind of sensitize themselves so that the whole hormone effect doesn't just shut off when the levels drop a little bit. The receptors are going to be increased on the surface. That's called upregulation. If the concentration of a particular hormone goes down, then goes up, then the cell will remove some of the receptors from the surface. Downregulation. So in other words, instead of all the cells being wigged out by this extra increase in hormone, they'll reduce their receptor number and kind of dampen out that effect. So upregulation and downregulation kind of keep the cell response a little bit more even keel when hormone levels go up and down. Finally, blood levels of hormone. This is the main mechanism of regulating hormone effects. Right? The body can regulate those hormone effects, and that's how it's done, by changing the blood concentration or level of the hormones. How is that done? It's done by changing the rate of synthesis and release of the hormone. Endocrine glands will be activated, release more hormone, blood levels will go up, we'll have more cell response. Negative feedback systems keep the hormone levels at approximately the same window range all the time, except under certain conditions when some hormones are designed to change the levels quite a bit in the circulation. So I drew a little cartoon to illustrate this principle. So here we see a population of target cells with hormone coming to them, diffusing from the bloodstream and binding to receptors. All those bound receptors are now activated, and the higher the concentration of hormone in the blood, the more that's going to come flooding over here, and the more receptors are going to be occupied. So that helps kind of make it a little more obvious. Something else cool about this slide shows that lipid-soluble hormones are not very good at dissolving in the blood plasma, which is water. In bulk, they're brought through the bloodstream on carrier proteins. They stick to proteins that have hydrophobic pockets, and that can help bring those steroid hormones throughout the circulation, and then they can dissociate to some degree and diffuse over to their target cells and do their thing. So we said that the main way that we control the blood concentration is by controlling the rate of synthesis and release of hormone from the endocrine tissue. And then so as we add more hormone to the blood, the concentration rises. Eventually, the hormones are going to be removed from the bloodstream in the kidneys. The liver is going to be involved in that too, in the kidneys. So how do we actually do that? How do we control the endocrine tissue so that it releases the hormone we want? There's three types of stimuli, three classifications of stimuli that can activate endocrine glands. And here they are, listed right here at the bottom. We'll come back to negative feedback. So I just drew in three little bubbles for different types of mechanisms by which the endocrine gland can be turned on. We'll start with a humoral mechanism. A humoral mechanism means a change in the concentration of something in the blood, and that in turn is going to activate the endocrine tissue. For example, blood calcium concentrations. We've seen in the past how important calcium is for signaling, and so its concentration in the blood is very tightly regulated. The parathyroid gland regulates calcium concentration. If calcium concentration goes down, this gland releases parathyroid hormone, which will bring calcium levels back up so that the blood level remains constant. Parathyroid hormone, it's called. Hormonal stimuli. Many times, endocrine glands are stimulated by hormones from another gland. So here we see an example of the hypothalamus actually releases hormones that trigger gland cells in the anterior pituitary gland. We'll come back to this. Hormones are released from the anterior pituitary gland, and they activate some additional glands that are shown here: the thyroid gland, the adrenal cortex, and the testes, and the ovaries. Those glands, in turn, then, of course, when they're stimulated, release their hormones. So that's called a hormonal stimulus that's coming down to these glands. And finally, a neural stimulus or neural stimuli. When sympathetic neurons, we can see them coming from the lateral horn of the spinal cord, when those sympathetic neurons are activated, they cause the gland cells in the medulla, the inner part of the adrenal gland, to release epinephrine and norepinephrine. So that's a neural stimulus. A neuron is releasing neurotransmitter that's going to cause those things to happen. The nervous system can actually override certain aspects or get involved with aspects of regulation of hormones. The sympathetic nervous system, for example, can cause blood glucose levels to rise. Normally, blood glucose levels are kept constant by hormones insulin and glucagon when you're at rest, but when you start exercising, hormones will come out that will start pushing up blood glucose, or at least maintaining blood glucose levels so that they don't drop as muscles start taking more glucose out of the blood during exercise. But all by itself, the sympathetic nervous system can elevate blood glucose levels, so it can override normal endocrine controls. So we have three mechanisms, three types of stimuli that can activate endocrine tissue. Increasing synthesis and release of hormones, it elevates the blood concentration or blood level. That causes more hormone to diffuse over and activate more receptors, and we get a bigger hormone response. What about getting rid of the hormones? If we want to stop activating a particular hormone function, when we stop releasing hormones, they are quickly inactivated, or at least the water-soluble ones. Here's a couple of mechanisms by which the hormones are removed from the circulation. In the blood, in the plasma, are enzymes that can break down peptide-based or amino acid hormones very quickly. As soon as we stop releasing peptide hormones, their levels in the blood go right down. The liver can add conjugate chemicals to steroid hormones. So steroid hormones aren't very soluble, but in the liver can add chemical groups on there to make them more soluble. Then they go back in the bloodstream. They're inactive when that happens, and now they can be released in the urine. The kidneys can get rid of them in the urine. So conjugation is another way of helping get rid of hormones that are inactivated. The kidneys and the liver, again, can remove hormones from the blood and get rid of them either in the bile or in the urine, typically in the urine. Here's an important definition. The half-life of a hormone is the time it takes for a concentration of hormone to drop to half of its original value. If we stop releasing the hormone and we measure its concentration in the blood, the half-life is the time it takes for the hormone to drop to half of its original value. That's the half-life. With peptide or amino acid hormones, that might just be in minutes. The half-life might be in minutes, whereas with steroid hormones, it might be weeks before the hormone drops down to half of its original concentration. So I'm just added in here. So our diagram is complete now. We have three mechanisms of stimulating endocrine glands, controlling the blood concentration, which then, of course, interacts with the target tissues, and then mechanisms of degradation. Now, as I said, the way we control blood levels is by controlling rate of secretion and release, because these removal processes are constitutive. Constitutive just means they're always going on. We're not regulating these processes. They're just always happening at a set rate, so we don't really worry about them or think much about them when we're talking about how we regulate hormone functions. It turns out that hormones have impacts on each other. Some hormones, such as thyroid hormone, that's a very great example, is a permissive hormone, meaning that thyroid hormone, when it binds to its receptor, increases the effect of other hormones on their target tissues. And without thyroid hormone, those hormones have less impact. So thyroid hormone, for example, increases the effects of epinephrine and norepinephrine on their target tissues. Right? If those hormones increase blood pressure, and you add a bunch of extra thyroid hormone, blood pressure will go up. Synergism refers to an idea that if two hormones each have a particular function or cause a particular cell response in the same target cells, and you release them both at the same time, the effect is greater than the additive effect would be. So if these two hormones together would produce a particular, if we added together their independent or individual degree of activation of the target tissue, and you put them together, there will be a greater activation of the target tissue, synergism. And antagonism, some hormones block the action of other hormones. Insulin and glucagon from the pancreas are antagonistic hormones, and we'll find out more about those when we talk about the control of blood sugar in those hormones. Well, we come to the point now where we're ready to talk about the actual hormones. That's all the background we need to understand pretty much the function of the endocrine system. So now we can look at the individual hormones. And so in order to do that, we're going to start out looking at an axis of regulation. The hypothalamus is like the top tier of regulation of much of the endocrine system, and it controls the pituitary gland, which in turn controls some other endocrine glands. The anterior lobe, there's two halves to the pituitary gland. Here's a diagram of the pituitary gland here, and you can see it has two lobes or halves to it. And the hypothalamus, in the case of the anterior pituitary gland, releases some neurotransmitters. Right here's a nucleus, a cluster of neurons in the hypothalamus, releases some neurotransmitters. They diffuse into this little cluster of blood capillaries, the primary capillary plexus, and now they're hormones, technically, right? A signaling molecule in the blood traveling to a remote location is a hormone. So then those hormones from the hypothalamus operate on the anterior pituitary gland and cause it to release these hormones down here. So we have a group of hormones up here that travel through the hypophyseal portal system, it's called. These connecting blood vessels, and wind up down here in the anterior pituitary gland and trigger the release of these hormones. And we'll learn those hormones as we go along. The posterior pituitary gland, on the other hand, is connected to the hypothalamus just by axons of neurons. The posterior pituitary gland isn't really a gland. It's just a collection of neuron axon terminals in the hypothalamus, but nevertheless, it's called the posterior pituitary gland oftentimes. So there's two hormones coming from the posterior pituitary, oxytocin, which you may have heard of, and ADH, antidiuretic hormone. They're both peptide hormones comprised of 9 amino acids. They're very, very similar to each other, but they have very different functions. Here's a diagram showing nuclei, clusters of neurons in the hypothalamus, whose axons travel right down and form the posterior pituitary gland and cause the release of oxytocin and ADH. Those are the two hormones, and we'll definitely take a look at those up close and see what they do. Oxytocin is the hormone that triggers uterine contraction during childbirth. Its regulation is by the cervix. As the cervix is deformed, that causes neurons to activate nuclei in the hypothalamus, and the hypothalamus releases oxytocin, which causes uterine contraction. That uterine contraction then pushes the baby head down onto the cervix, stretches it more. The more you deform the cervix, the more oxytocin keeps coming out. We get higher and higher levels of oxytocin, a positive feedback system, until the child is born. Oxytocin also causes milk ejection, so milk production in the mammary glands is triggered by hormones, different hormones, prolactin, and then oxytocin helps bring that milk to the point of ejection so it can be available for the infant. Interestingly, more recently, oxytocin has been discovered to be an important neurotransmitter in the brain, involving our ability to empathize with other people, to read their emotions and empathize with them, form bonds, emotional bonds with each other. Okay, lastly, antidiuretic hormone. It's a peptide hormone that responds to dehydration. When you get dehydrated, you're losing water, say by sweat, from the blood, and the blood is becoming more concentrated. Osmolarity refers to the concentration of the blood plasma, the solutes. For losing water out of the blood, things are getting more concentrated. The hypothalamus has osmoreceptors, cells that shrink by osmosis when they find themselves in this hypertonic environment, right? There's increasing concentration of solutes, and that causes the release of antidiuretic hormone. The other stimulus that the hypothalamus responds to is a drop in blood pressure. If your blood volume is going down because of dehydration, loss of water, the blood pressure also goes down. And we need to come back to that in chapter 19, but that's just a way things work. If blood volume goes down, blood pressure goes down, that causes ADH release. What does it do? Antidiuretic hormone. Well, the term diuresis means production of urine. Antidiuresis, or antidiuretic hormone, reduces or slows the production of urine. If you're already dehydrated, the last thing you want to do is pee out more water, and so you're going to retain water in the bottom by the action of this hormone. Get dehydrated, reduce the urine production. Actually, interestingly, ADH is a powerful vasoconstrictor. Now again, we're bumping up against chapter 19. That just means that blood vessels are going to contract the smooth muscle in their walls, and that's going to also drive up blood pressure. So we see that one of the features of dehydration is a drop in pressure. We're going to bring that blood pressure back up. So those are the effects of antidiuretic hormone. Elevate blood pressure and reduce the osmolarity of the plasma by causing us to retain pure water that would have otherwise been peed out in the urine. Incidentally, extreme pain can cause the release of antidiuretic hormone, which will drive up your blood pressure. If you ingest ethanol, ethanol will inhibit ADH release, and so when people consume ethanol, it causes them to have a very highly increased rate of urine production. People that drink alcohol will be peeing a lot because their ADH has now been reduced, and so instead of antidiuresis, we have diuresis. There are drugs, though, that very importantly help us get rid of excess fluid from the body. There are important times when we want to pee out some water, and we'll talk about that when we talk about the kidneys in chapter 25. But diuretics are drugs that increase the production of urine, and so that'll override the effects of ADH. There's actually a condition in which there's too much ADH produced. It's called the syndrome of inappropriate ADH secretion, and people that have that problem, they have too much ADH, not enough urine production, too much higher than normal blood volume, and increased blood pressure, hypertension, which can cause headaches, fluid retention, edema, and various problems. Some tumors, some cancer cells, interesting, this happens in other cases, but some tumors produce hormones. They're just cells that don't do what they're supposed to do. They act abnormally, and one of the potentially abnormal functions is to release hormones. Some lung cancer tumors release antidiuretic hormone, which can cause all kinds of problems as the blood volume rises too much. Some people don't have ADH. They don't produce ADH, so they have a very elevated rate of urine production. Diabetes insipidus, you see that familiar term diabetes. There's a high rate of urine production and a lot of dehydration. If you're peeing out a lot of volume, you're always going to be dehydrated, got to take in a lot of water to compensate for that. So people with diabetes insipidus have a tremendous amount of urine production and a tremendous amount of water intake. All right, so that's quite a few topics. We got a lot under our belt already in understanding the functions of the major control system of the endocrine system, the hypothalamus and the pituitary gland. We'll come next time back together to talk about the anterior pituitary gland and some hormones that are controlled by the hypothalamus in that part of the gland.